Hello everyone, we are the Masters of Physics, and today we'll be presenting on Foundational Concepts 1 and 2, with some focus on Foundational Concept 5. Alright, so here's a preview of the content categories. First, let's review functional groups. As you can see, we have hydrocarbons. These are just carbons and hydrogens. When we add groups like NH2, then we can have things like amine, or we add OH to have alcohol. When we introduce carbonyls into the equation, then we can get groups like carboxylic acid, which is a carbonyl group and an OH group, or we can have derivatives of carboxylic acids, such as esters. So that's a carbonyl with an OR group, or we can get something like an amide, which is a carbonyl with NH2. And this will be important to recognize in amino acids. Okay, so hopefully you're able to recognize the functional groups on here. So if we take a look at the image on the left, and we look at the center, we're going to see the alpha carbon bonded to an amino group, which is located to the left of it. And then there's a carboxylic acid group to the right of it. And then on top, we have the R group, which in most cases will determine the property of the amino acid. And as you can see, sometimes the amino group can be protonated and the carboxylic acid group deprotonated. And when this exists, we see the Zwitter ion, which is basically the neutral form of the amino acid. So for the MCAT, it'll be important to know 20 amino acids, which are broken into nonpolar and polar amino acids. Polar amino acids are further broken down into non-charged, charged, or acidic, and basic amino acids. And keep in mind that most of these amino acids will be L configuration, so the amino group will be found to the left. So amino acids are joined together by peptide bonds, which are very stable because of their double bond character, and this results because of the resonance. To break this bond, you have to use hydrolysis, which can include acid hydrolysis, which is non-specific, or prokaryolysis, which cleaves specific bonds. So, when these amino acids come together via peptide bond, they form polypeptides, and this is what is known as the primary sequence of a protein. The secondary sequence arises when we have backbone interactions, so this is with hydrogen bonds that form alpha helices and beta sheets. Then we can have additional interactions such as hydrogen bonds, van der Waals interactions, hydrophobic packing, and disulfide bonds, which result because of the cysteine residues, and this gives rise to our tertiary structure. And then finally, we can have different subunits interact with each other, and so this would result in a quaternary structure. Alright, so now we're going to take a look at how we can exploit these properties we just learned about in order to separate and analyze proteins.
5C, separation and purification methods. The big picture here is this category covers useful techniques to separate and purify proteins. These methods include extraction, distillation, liquid and gas chromatography, and electrophoresis. Depending on the types of substances which comprise the mixture and the strength of the intermolecular interactions will determine which methods you use. Extractions separate compounds using immiscible or unmixable solvents using a separator funnel. The compound of interest may be found in a polar aqueous layer or nonpolar organic layer. Distillation used to separate substances in a liquid based on their boiling points. The liquid that has the lowest boiling point will be vaporized and collected first. Gel electrophoresis is an experiment that's used to separate different components of a mixture based on the size and charge. SDS-PAGE is an example of a detergent that denatures proteins and adds negative charges proportional to the size of that protein. Keep in mind, if you want to separate proteins just by their size or the number of amino acids, use SDS-PAGE. Quantitative analysis encompasses anything that allows us to determine the protein concentration. Enzyme-linked assays is used to detect antibodies or antigens. Bradford assay, for example, a negative charge blue dye binds to positive chains of proteins to form a blue complex, and UV spectroscopy, the wavelength absorbed by the sample tells us the presence of the compound. Chromatography is a technique used to separate mixtures by their individual components in order to analyze, identify, purify, or quantify the presence or absence of a component of a mixture. Components are carried through a stationary phase by a mobile phase. The stationary phase is fixed in place and could be a solid or a liquid. The mobile phase that carries the elution could be a gas or a liquid. Thin layer chromatography, or TLC, is useful in separating amino acids and small peptides based upon the differences in polarity by determining the distance of the nonpolar molecules traveled away from the polar origins through capillary action. Additional chromatography techniques include affinity chromatography, gel filtration or size exclusion chromatography, and ion exchange chromatography. Affinity chromatography targets a specific substance using a stationary phase with a high affinity for a specific substance. Gel filtration uses beads with small pores to separate molecules based on size. Ion exchange uses positively or negatively charged stationary phase to attract anions or cation molecules, respectively.
So a brief overview of fermentation. We have first glycolysis, which produces two pyruvate, and those two pyruvate either undergo fermentation to produce lactic acid or ethanol, depending on alcohol fermentation or lactic acid fermentation. And one of the most important things in fermentation is that NADH is produced, which is recycled from NAD+, which can then go through glycolysis again to produce more ATP. NAD+ and FAD are both redox cofactors, which means that they are helpful in redox reactions within biological systems, and they help the enzymes that undergo redox reactions. Therefore, they could reduce to NADH and FADH2. Another interesting thing about these two is that they're electron carriers. And electron carrying is very important in specifically aerobic conditions. And electrons move from low reduction potential to a higher reduction potential. Glycolysis uses redox reactions and creates electron carriers to use in the electron transport chain in aerobic conditions. However, in anaerobic conditions, which was mentioned previously, NADH is recycled to NAD+, so that glycolysis can be repeated to maximize ATP production, since ATP cannot be produced in the citric acid cycle and the electron transport chain. So I want to briefly overview aerobic conditions, or the electron transport chain, in which electron carrying goes from complex 1 into coenzyme Q, in which complex 2 also goes into complex Q. By the way, complex 2 is also succinate, which is in the citric acid cycle and is an intermediate. And then it goes to complex 3, cytochrome C, complex 4, and oxygen converts into H2O. Something that's also important, that's also a redox cofactor, is NADPH, which helps with oxidative stress within the electron transport chain and is produced by the pentose phosphate pathway, which is one of the important products of the pentose phosphate pathway. It is also very important to mention that redox cofactors occur in both beta oxidation, which is fatty acid breakdown, and fatty acid synthesis. And you can see the redox reactions occurring within the mechanisms by what is circled. So we have NADPH being converted to NAD+ in fatty acid synthesis, and we have FADH and NADH2 being produced in beta oxidation.
So a prokaryote is an organism, a single-celled organism. Most tend not to have a nucleus, but some might have a distinct nucleus, and it includes bacteria, archaea, and cyanobacteria, but it's two important domains of the bacteria and domain archaea. For cyanobacteria, it's known to be a very important evolutionary bacteria that produced atmospheric oxygen and also had the ability to remove atmospheric nitrogen and before human beings existed. So the domain bacteria or singular bacterium is important to our existence. It's important for our existence. Bacteria makes up about 100 trillion cells within our body. We have digestive bacterial guts that help in our breaking down foods. Bacteria reproduces fast through binary fission, where a circular chromosome splits into two. Unlike eukaryotic reproduction, bacterial reproduction has cell replication and cell separation happening at once. Bacteria have like cell walls made of peptidoglycan, which is like a linked sugar layer, which gives it a rigid structure. And bacteria moves with flagella or sometimes pili, which they use for attachment, and fimbriae as well, what they use for attachment or for bacteria locomotion. Bacteria has very distinct shape. They can come in spheres or coccus. They can come in rods or bacilli, or they can come in wiggly, squiggly structure or spherochates. And to identify whether a bacteria is positive or negative, you stain it and analyze it under a microscope. So when you put on the crystal violet and it comes out purple, that means the bacteria is a positive bacteria with a peptidoglycan wall. But when you put the purple violet and you were able to wash it off and put iodine and then saffron and it turns red or pinkish, that means it's a gram-negative bacteria. So bacteria like E. coli is a gram-negative bacteria, and they tend to be very dangerous because they have LPS, and that makes it like able to like resist antibiotics and host human cells. Another importance of bacteria is its ability for genetic recombination, where it carries plasmids and DNA and into a different organism, way different from its parent organism, and is able to be used in a whole lot of genetic researches.